Good afternoon, my name is Ekaterina Mironova. I am PhD, head of the Laboratory of Molecular Biology of the Cell, St. Petersburg Institute of Bioregulation and Gerontology, and also head of the Laboratory of Molecular Neuroimmunoendocrinology, St. Petersburg Research Institute of Physiopulmonology. Today I will tell you about molecular mechanisms of pancreatic function peptide regulation. The pancreas is an important organ of the digestive system, necessary for the nutrients absorption. It performs exocrine and endocrine functions. The pancreas exocrine functions is realized by the pancreatic juice secretion, which contains enzymes involved in digestion. The pancreas endocrine function is realized by the production of hormones, insulin, glucagon, somatostatin, and gastrin, involved in the regulation of carbohydrate, fat, and protein metabolism. In recent decades, the incidence of type 2 diabetes mellitus and chronic pancreatitis has been steadily increasing. According to the World Health Organization Expert Commission, more than 130 million people in the world currently suffer from diabetes. This figure increases annually by 5 to 10%. The increase in the diabetes prevalence is primarily observed among older people. Chronic pancreatitis is also a common disease of the pancreas. The incidence rate has doubled over the past 10 years. This is associated not only with improved diagnosis of the disease, but also with the increased impact of unfavorable environmental factors, as well with the increase in the average life expectancy of the developed countries' population. One of the reasons for the age-associated development of type 2 diabetes mellitus and chronic pancreatitis is a decrease in the number of pancreas, acinar, and islet cells. At the subcellular level, this is associated with a disruption in the expression of signaling molecules, markers of the pancreatic cell functional activity. In this regard, the development of new drugs used to restore the functional activity of pancreatic cell is becoming increasingly socially significant. Substances of natural origin are of great interest given their safety and lack of toxic effects. Such preparations are peptides created at the St. Petersburg Institute of Bioregulation and Gerontology. Based on the analysis of amino acid composition of polypeptides complex supraport, which was isolated from the pancreas of young animals, the tetrapeptide KEDW was synthesized. It has been established that with pancreatic cell aging, the differentiation markers expression decreases. Tetrapeptide KEDW stimulates the expression of acinan differentiation factors and islet pancreatic cells in young and old cultures. The islet and acinar pancreatic cell differentiation induced by the tetrapeptide KEDW may be one of the mechanisms of its anti-diabetic and anti-inflammatory actions. It was found that the tetrapeptide KEDW also increases the expression of metalloproteinases, serotonin, glycoprotein, anti-apoptotic protein, proliferation factors, and reduces the expression of pro-apoptotic protein in old pancreatic cell cultures. Thus, the tetrapeptide KEDW activates the expression of signaling molecules, markers of the pancreatic cell functional activity. The KEDW tetrapeptide effect on the pancreas functional morphology in rats with diabetes mellitus modeled by streptozotocin was studied. In the group with experimental diabetes mellitus, the insulin-producing B cells overage area decreased under the streptozotocin action, whereas the glucagon-producing alpha cells overage area increased. The use of the KDW peptide led to the pancreas reparative and compensatory processes development, which was manifested in an increase in the insulin expression by the Langerhans islet B cell and a decrease in the glucagon production by alpha cells. The KDW peptide effect on the pancreas endocrine function was studied in laboratory primates, female rhesus macaques. A 10-day course of the KDW peptide resulted in a decrease in the insulin and C-peptide concentration plasma rise in most all animals 5 and 15 minutes after glucose standard dose administration. The peptide also resulted in the increase in the rate of glucose disappearance from circulation to the liver seen in young animals. The KDW peptide restorative effect on pancreatic function was also partially maintained 3 weeks after drug withdrawal. It was found that Vasogen activates reparative and regeneration processes in the old rat pancreas in the radiation aging model. Vasogen has a pronounced effect on the pancreas microcirculatory bed, which was expressed in the capillary architectonics normalization and a decrease in vascular congestion. The use of endolutein in patients with type 2 diabetes mellitus resulted in a decrease in glycemia, glycosuria, and the glycosylated hemoglobin level in the blood. In individuals with hypertension manifestation, arterial pressure normalized and the heart muscle diastolic function improved. Endolutein also had a long-term normalizing effect on carbohydrate metabolism, which is modulating and decreases as disease compensation is achieved. The use of endolutein led to the atherogenic lipid fraction content normalization in the blood. Peptidergic regulation of pancreatic function is achieved through the pancreatic peptides' influence directly on the islet and acinar cells' functional activity. Vascular peptides on the microcirculatory bed state, as well as the pineal gland peptide stabilizing effect or on carbohydrate metabolism and neuroendocrine regulation optimization of organ function. Thus, for the pancreas functional activity disorders correction of various origins, it is advisable to use the peptide preparation Suprifort, Vasogen, endolutein in combination. Thank you for your attention.